Long before anyone named cellulase, cellulose was already gathering here boll by boll, row after row. In ripening cotton, the molecule becomes a landscape. From cotton fields to these mountain forests, the same quiet architecture rises into trunks, ferns, and a canopy spread across the valley. Cellulose gives the landscape its lift, holding green life upright in the morning light. Even here, stacked by the river and weathering in the open air, those fibers remain stubbornly bound. Cellulose backs its chains like cordage, giving each log a strength that does not easily yield. Inside the fiber, water glints and slips through translucent passages, while the brown strands hold their chains tight. To ordinary chemistry, the sugar is still behind a wall. But here on the rain-dark forest floor, nature has been opening that wall all along. Pale fungal fans spread over fallen wood, while unseen microbes help turn hard plant remains stored new soil. Out on the savanna, the same hidden labor moves through waving grass and grazing herds. Inside each animal, tough plant fiber enters a wider society of living digestion. The first human clue came not from industry, but from curiosity. In 1838, the French chemist Anselme Payen isolated the tough plant substance and gave cellulose its name. Payen's work separated cellulose from the confusion of plant matter. It made the material visible to science, even while its undoing remained mysterious. Through the 19th century, observers watched seeds soften their own cell walls, and fungi eat what seemed inedible. The idea of a cellulose-cutting ferment began to take shape. The clues were humble softened straw, rotting paper, damp cotton, and the patient appetite of molds. Around the turn of the 20th century, researchers including C. Salyer studied digestive fluids from snails and showed they could attack cellulose. In damp plant tissue, pale fungal threads probed the honeycomb of cell walls. The name cellulase came to gather around this power. Not one tool, but a family of enzymes opening plants from within. Then history took an unexpected turn in war. In the humid Pacific, cotton tents, uniforms, and rope began to fail under an invasion too small to see. Military scientists collected the damaged fabric because loss had become evidence. The enemy on the cloth was a fungus with an extraordinary hunger for cellulose. One isolate, later known as QM6A, came from that world of ruined cotton. It would become the ancestor of a major industrial cellulase lineage. After the war, Elwin Rees and colleagues studied such fungi with uncommon patience. The organism was later honored in the name Trichoderma. From that patient work, cellulase entered a wider landscape, fields, tanks, and green buildings shaped around working biology. Its promise was nature's own, taking plant abundance apart without violence. Inside the wall, the work is intimate. Small enzyme forms find the grain of cellulose and settle along it. Fibers loosen into drifting threads, and tiny sugars pass through the light. Along these tangled corridors, some clusters open the cellulose chains from within. Others gather at the curling ends, drawing off shorter fragments. In the golden flow, the last enzymes finish the work, loosening short fragments into glucose. Tiny sugars stream away from the dissolving fiber. The simple form plants once built from sunlight. Here among amber crumbs and pale fungal threads, that grace is almost visible. Enzymes seep into softened wood and begin only where they fit. No force, just a precise touch, opening the fiber from within. From wood, that same quiet logic moves into valleys of field and cloth, where river water and plant fiber meet patient craft. In such places, cellulase offered a gentler way to refine fibers and soften harsher treatment. Across these long runs of cotton, cellulase could work at the outermost fibers, trimming the tiny fuzz that catches against the hand. What remains is cloth that moves more cleanly and feels softer, a lesson mills would soon carry into denim. In denim yards like this, blue cloth moves through water, troughs, and open air. Cellulase gave finishing another tool besides rough abrasion. At the cotton surface, biology could help soften the color and the hand. Beyond the mill, the same quiet chemistry followed cotton into everyday washing. Across rooftops and courtyard lines, white sheets lift in there while cellulase works unseen at the fiber surface, easing away the fuzz that makes cloth look tired. Back at the mill, forest, river, and steam frame another use for the same enzyme. Cellulase can help pulp fibers partandre more easily, bringing a biological nudge to world once driven by force. Here, the same idea follows. The harvest into the orchard yard, crates of fruit move toward the press house, where cellulase can help pulp let go of its juices. What clung together begins to open, and the flow runs clearer. Across the harvested fields, straw lies in long amber rows, with chaff lifting in the low light. In this rough plant matter, cellulase can help loosen the fibers that make stalks and husks so stubborn. Here beside the market gardens, dark windrows breathe in the morning light, releasing the warmth of decay. In that slow turning, cellulase helps spent leaves and stems soften back into living soil. Out here among wind-bent test plots, access roads, and distant silos, the promise stretches wider, fuels and chemicals drawn from residues already left by farming. Cellulase stands at that doorway, helping open dry stalks without tasking food fields to become fuel fields. Inside the straw, cellulose looks less like a powder than a fortress of layered walls and splintered fibers. Along those ridges, enzyme partners gather and work the openings, turning resistance into a path. In the wet tangle of decaying grass, fungal threads lace the gaps while bacterial clouds drift through golden droplets. Cellulase belongs to this larger chorus, one voice among partners answering the next. From above, that chorus widens into fields, stubble, hedgerows, and the pale curve of a river. As the harvest moves across the slope, fibers are taken, residues left, and unseen enzymes help keep the circle turning. Here old trunks and new saplings share the same valley, while fallen wood begins its quiet return to the soil. Cellulase belongs to the ancient craft, not new machinery, but biology borrowed with care. Beyond the reeds, fields and quiet truths gather at the wetland's edge, a landscape learning to work with what grows. The aim is simple: waste less of plant abundance, and let biology do the finer work. Here, where river, reed, and orchard meet the morning light, that idea feels almost visible. Cellulase is one way biology opens plant fiber gently, turning what was bound into something useful. At Enzymes Bio, we're a small team of scientists and biotech enthusiasts. We bring the world's enzymes onto one platform so startups and labs skip the procurement maze. A greener chemistry for the years ahead. Learn more at Enzymes Bio.